Okay, guys, so we talked about all the cell organelles in our last lecture, and this time around we're going to talk about one specific organelle, okay, and that is the cell membrane. And the whole reason that we're going to talk about just that one is because the cell membrane is really kind of the thing that controls what comes into and what comes out of the cell. So if I had to say that there were two of the most important organelles in the cell would be the nucleus, which tells the cell what to do, and the cell membrane, which tells what can come in and what can come out. Okay, so in your notes you should have what is the job of the cell membrane. Okay, that is to maintain homeostasis. Oh wow, that's spelled wrong. Maintain homeostasis. Okay, and what that means is it's going to control a balance. Okay, we're going to keep this stuff on the inside of the cell and this stuff on the outside of the cell in balance. Okay, and we're going to do that by controlling what enters and leaves. So, if that was confusing to you, the job of the cell membrane is to maintain homeostasis by controlling what enters and leaves the cell. Okay? Alright, so you should have a picture of the cell membrane title in your notes. Okay, this is what I want you to draw. A blob, layers of balloons, another blob, layers of balloons, another blob with a line going through it, kind of like a slip and slide. Okay, another row of balloons, and a blob. Now this blob, I want you to draw almost like a flag. Okay, coming off of it. Alright, so pause the video if you need to, draw that picture. Okay, now, what is the cell membrane made of? So underneath your picture of the cell membrane, I want you to draw a dot that says made of proteins and lipids. Now, the balloons are the lipids, so you're going to need to label that in your picture. The blobs that you drew are the proteins. Again, label that in your picture. Now that is going to be a quiz question. It's also going to be a test question, so you need to make sure that you're taking care of business and getting this picture drawn. Okay, so again, the proteins are the blobs, the balloons are the lipids. Alright, there are three types of proteins. Okay, you should have those listed in your notes. The first one is a receptor. Okay, so a receptor protein is going to be just a regular looking blob. And what its job is to send and receive information to and from the cell. Okay, so maybe the cell needs to let something in, it needs to let something go, maybe the cell's about to die. Okay, that is what a receptor protein is for. We actually have receptor proteins in our body too that tell us things like, are we hot? Are we cold? Did we just burn ourselves at the stove and we should feel pain? Okay, so all of those things, we have those too. So again, receptor proteins. Okay, the second type is the one that looks like it's got the slip and slide through it. That's called a channel protein. Now what a channel protein does is basically kind of like a slip and slide. It gets you from one place to the other quicker. Okay, so you can't slide through the grass, or you can, but it hurts. Okay, slip and slide helps you slide from one end of the grass to the other, right? Think about this sucker right here. Okay, this is the grass. You're standing right here and you need inside, you're going to use that slip and slide to get inside the cell. Okay, and the last type of protein is called a marker protein, and that's going to be this flag. Okay, the protein that's got the flag coming out of it, and basically what those do is identify the cell. Okay, so that's going to say, hey, this is a blood cell, hey, this is a liver cell, something like that. Alright, now you've got a T-chart. Okay, the left-hand side should say active transport, the right-hand side should say passive transport. You need to know a couple of things here. Passive transport requires no energy. Okay, so that should be your first empty bullet point. It requires no energy. Okay, and then in passive transport, we move from high concentration to low concentration. Okay, so what that means is you're going to have a whole lot of things in one area, and they're going to disperse out among the cell, okay, until they equal out or reach equilibrium. An example of this would be diffusion, also osmosis. Now you don't actually need to write down anything about diffusion or osmosis yet. Save that for a second. I want to show you guys a couple of examples. Alright, so basically what happens here, this would be diffusion. Okay, we've got a whole lot of particles sitting right here. Now, okay, what would happen if we, say this was perfume, we sprayed this as a bottle of perfume. Okay, what would happen, that perfume would eventually diffuse out and try and fill up this whole space, okay, with an equal amount of perfume particles. I'll show you an example here in just a second. Alright, so here's just kind of an example. Watch the green particles of what happens with diffusion in cell. Alright, so did you see how the green particles kind of moved in and out to try and reach equal numbers? Okay, that's exactly what would happen. Now, I need you to know, okay, what you do need to know about diffusion. Okay, diffusion is the movement of particles. Write that down. Diffusion is the movement of particles. Okay, now that's different from osmosis, which is the movement of water. Okay, and I'll show you that here in just a second, but get that written down. Osmosis is the movement of water. Okay, so again, osmosis is just a type of diffusion, but it is the movement of water. Now watch the Mickey Mouse heads. Okay, here's your cell membrane. Watch what happens with the water. Okay, so you see how they're trying to move to be equal? Okay, it's not perfect, but it's attempting to move that way to make it equal. Okay, now what happens if you cannot get into the cell? Okay, maybe you're a particle that's too big, like these red particles. Okay, that's why we use those slip and slides, those channel proteins. Okay, so again, keep an eye on the red particles and see what happens. Okay, makes sense. They're using those channel proteins, those slip and slides, as a way to get into the cell. Now, in this next picture, okay, in this next slide, I need you guys to explain to me what's happening. Alright, now watch everything that's happening. Okay, I need you to explain to me what is going on in this picture. Okay, so I hope somebody said osmosis of the Mickey Mouse, diffusion of the green dots, and facilitated diffusion of the red particles. Okay, that's what I'm hoping. Alright, now you should have drawn 3 little beakers with some water and some circles in them. Okay, 3 different size circles. And it said passive transport in the cell, I hope. Okay, now underneath that, I need you to write 2 words, solute and solvent. Okay, because these are not in your notes, but they're very important. Okay, so a solute is going to be a gas or a solid. Now, a solid could be something like salt or sugar, but it's some sort of particle. Okay, the solvent is going to be the liquid that particle is going to get dissolved in. Now, mostly our solvent is always going to be water. Okay, so I need you to write that down. The solvent is usually always water, and that's going to make your life make a lot more sense here in the next few minutes. All right, so we've got 3 beakers. Okay, this is going to be a skin cell right here that we put in each of those beakers. And you actually drew those skin cells, but yours look a little bit different. Okay, now, this right here is going to be in 100% water. The cell inside of it is 80% water. Okay, cell in the middle, we've got 80% water outside the cell, 80% water inside the cell. And then in this last beaker, we've got 70% water outside the cell, 80% water inside the cell. Okay, so pay attention to what I'm about to write on the board right now to make that make more sense. Okay, based on your beakers in your journal and what I just wrote down, go pay attention to the board. All right, now I'm hoping the passive transport makes a little bit more sense to you after what I just explained on the board. Okay, now we got to come back and fill in the rest of our T-chart about active and passive transport. So 2 bullet points. First thing you need to know, active transport requires energy, and molecules are going to move from low concentration to high concentration. So basically they're pushing against what they know they should. Okay, that's why they require that energy. Now there's 2 types of this. The first one is called endocytosis, and basically what endocytosis is used for is bringing stuff into the cell. And the last one is exocytosis, and that's taking stuff out of the cell. Now, this is a confusing topic, but we're going to practice it for like the next 10 days, and you'll be good by the time we're done. I promise.